Our last speaker of the session is Martin Tresguerres from the Scripps Institute of Oceanography at UC San Diego. And the talk is the critical importance of external and internal microenvironment on the neurobiology of marine animals.
Well, hi everybody. Thanks to the Cowley Foundation and the Allen and the Paul Allen Frontier Group for organizing this and inviting me and all of our group. It's been really great day of sessions. And today I'm gonna just, I think this also, thank you to the organizer because I think this session was very good. It complements all the topics each other very well. And today, what I'm going to talk a little bit now is about thinking a little bit about, we heard a lot of really cool and sophisticated talks about neurobiology, transgenic animals, recordings. One time I tried patching cells during my PhD, and then I decided to study osmoregulation. So I respect this field a lot. But then trying to integrate the neurobiology within the broader concept of both in the environment, in terms of marine environments, it could also apply to terrestrial environments, but also the environment that based on where the neurons are located. Now, the idea when we try to focus on changing environments and climate change, the choice of focusing on the nervous system was great. Thank you, Angie, for spearheading all this group, all this research, and it's essential because it's important for many functions, as also is an interface with the environment. However, some of the things we need to take into account is that the nervous system is not in contact directly with the environment. There's a lot of, it might be, in the case of vertebrates, inside the blood-brain barrier, many of the cells, not all of them, but many of them. There are blood that maintains homeostasis or not. And all these systems, even though it's not directly neurobiology, these integrative homeostatic responses are essential for neuronal functions and also for determining resilience and vulnerability to changing environments. Now, going into the other direction, and this is one of the big advantages of being at Scripps Institution of Oceanography, we interact a lot with people who are measuring the environment in really unique, both temporal and spatial resolution. And this is a study that I read about 10 years ago now, maybe more, 2012. Christina Frieder, she was a PhD student in Lisa Levin's lab. I was on her committee. And she was using some of these sensors developed by Todd Martz, who's in our Allen Center. And what I'm showing there in the right are two sensors, that's called CFOX, C, pH, oxygen, so CFOXes that are placed in the kelp forest right outside of Scripps, and they're put at two different temperatures, two different depths. And when you look at dissolved oxygen, pH, and CO2, we see that normally this is a naturally fluctuating environment. We see those in kind of like in orange, you see those like rapid changes in oxygen and pH and CO2 that are due to photosynthesis and respiration. So during the day, there's more oxygen than compared at night because the balance of photosynthesis and respiration favors more oxygen. And then we can also see more pronounced swings, which are based to other oceanographic conditions such as upwelling or currents. And also you can see there how the gray line is, I believe, at 7 meters depth, where the purple line is at 17 meters, both in the same location. And you can see that great variability of all these parameters. And the animals are living there, and not only they're living there, but the animals, microbes, and algae and plants are actually creating these disturbances themselves. So when we take into account the measure, like these natural ranges that happen, as I mentioned, those bigger dips are by upwelling. You can see how dissolved oxygen can change, and you can pick your favorite unit, but from hypoxic or nearly hypoxic conditions to hyperoxic conditions. pH can change in this case, between 7.6 and 8.2. The 7.6 is way lower than any future ocean acidification scenario. And CO2 also in the opposite direction. And similar things happen when we look at temperature, and also they correlate with pH and dissolve oxygen. But these changes in temperature, this is over a period of about 2 months. You can see how there are temperature changes of about 10 or 12 degrees that happen both on a kind of like night-day basis as well as a seasonal basis. And animals are living there, and this goes to the concept that the nervous system is one of the main things is to adapt to these changing environments that happen naturally. That's something that is very important to keep in mind when we're looking at responses to human-induced climate change. Now, based on that, I want to do that. I want to measure pH and oxygen in different places. So we got some of those sensors from Todd, and here I'm the one with the yellow snorkel with my colleague David Klein in Panama. And over a couple of years, we did measurements of pH, oxygen temperature, salinity, and other things in coral reefs, but we did it in the 2 different depths and also in different reefs. You might have a reef that is more like you would see in the Discovery Channel, the pristine reef, very transparent with a lot of flow, but then other reefs are more sheltered, more like a lagoon. And we saw that, and this is other people have seen this as well, that there's also that very pronounced variability in oxygen and pH and CO2, which is due for photosynthesis, respiration, and calcification. Differences between depth and also very pronounced differences in terms of different types of reefs. So again, going to the concept that when we look at the external microenvironment or the environment at the level where the animals live, it's a lot more variable than we maybe consider a few years or decades ago. And then because most of these disturbances are actually being generated by the animals and then the algae and the plants, the closer we get to these sources of variability, the more pronounced that these changes may be. In this example, in addition to our sea foxes, we just went and took sample from what is labeled as DS. We call them like damselfish shelters because we were studying responses of damselfish to ocean acidification. And a little bit to our surprise, we found that this is during the day, the pH and the CO2 in those shelters, which is where the animals live most of the time, not all of the time, but most of the time, it is a lot more acidic and a lot more hypercarbonic than what we expect from ocean acidification in the open ocean in 100 years. And when you see in the video in the right, that's what the real world looks like. That is, I'm gonna try to play it again. So this is like, no, fish don't live in tanks. They don't live in containers under perfect conditions. They're all hovering there in this coral reef. They're like shelters, crevasses, different environments, different shades, different things moving all the time. And the nervous system and other homeostatic systems are tuned to that. And that is something that I think is a very cool and important thing that over the past several years, we're starting to return to studying these conditions with more like biomedical grade techniques. It's a lot more complicated. It's a lot more difficult to get those like asterisks that allow us to publish in cool journals, but it's absolutely essential and I would argue more fascinating. Now, if we take a look specifically at the problem of ocean acidification or high CO2, it's sometimes we cannot always do super cool transgenics. Sometimes we have to go to basic physiology. And something that is very important, and this is perhaps the best example about how the internal environment can be very different from the external environment, is the process by how fish and most other animals are able to regulate their blood pH. This fancy diagram in the left is called a Davenport diagram. It has 3 axes. You have pH, bicarbonate, and then the blue lines are isopleth for carbon dioxide. And the dotted line is the non-bicarbonate buffering line of the blood of this animal. So if you imagine in a controlled condition, the fish is producing CO2 due to its metabolism. There's a lot more CO2 inside of the fish than outside, regardless of except for very, very specific conditions like aquaculture or some CO2 seeps. In the vast majority of cases, there's more CO2 in the fish than outside. And that determines that the CO2 diffuses out when it goes, the blood goes through the gills, but then inside of the animal, the CO2 is in equilibrium with pH or with bicarbonate and with hydrogen ions or pH. Now, when you increase carbon dioxide outside, what happens is that the CO2 inside builds up until it reaches a certain partial pressure difference that it can diffuse back again at the same rate. But then as a result, that CO2 generates more protons and more bicarbonate, and that initially lowers the pH of the blood. However, the gills, they become activated and they excrete acid actively through these cells. And then as a side effect, there is further accumulation of bicarbonate inside of the blood of the fish. Now, why is this relevant to neurobiology and other things? Well, you might have heard that one of the potential effects of ocean acidification is on fish behavior, and the hypothesis behind that is that when you have more bicarbonate and maybe a little bit less chloride, there might be a slowdown or a reversal of GABA-A receptors that turn GABA-A receptors from inhibitory to less inhibitory. And that has been proposed to be one of the main causes leading to, for example, olfactory malfunction and other things. Now, when I told this story to my colleague Trevor Hamilton, who's also in ADC NICE, he's a neurobiologist by training, working with mice and human slices, he was like, well, if that was the case, then you would expect that reversal of GABA-A function would result in anxious fish. I was like, what? How do you measure anxiety in fish? And it turns out that there's these very standardized methods that work with rodents, but also with fish, such as the light dart test. Many of you might be familiar with that. Normally the fish, when they're not anxious, they explore this arena that has half of the wall black, the other one white. And that video there, which is sped up about 4 times, you see in the top, a fish under controlled condition is exploring the box, whereas the other one, which has been exposed to CO2 of about 1,000, maybe 1,200 microatmospheres for a couple of weeks, is just not moving. It's kind of very hesitant. It even looks anxious there. And eventually it will like go and crawl to the corner and stay there. And we did this with just automatic tracking software, and this is a representative heat map clearly showing that there is something going on there. The CO2 fish, they are really literally crawling into the corner, and you can see the anxiety there. And we repeated that with many fish. There we have zebrafish, rockfish, the damselfish from Panama. More or less, on average, all the fish respond the similar way. We can modulate this with GABA-A receptors agonists and antagonists in the way that you would predict. But something very important here is that if you see in the left are control fish, we also try dopamine agonists and other things. But the one thing that many times we try to ignore, but I think here it might be very, very important, is that not all fish respond exactly the same, and they do not all respond to the same degree. And that's why we have these like error bars. And where that might make it more complicated to publish a paper, that is very, very important, because that shows that not all fish do the same way, which has to be related to something that provides that variability over which natural selection can take place. Now, another thing that also we have going more into what are the neurons and how the GABA-A receptor might be happening, because I told you the responses in the blood. However, neurons, most of the neurons are not exposed to the blood, but they are inside the brain, enclosing the blood-brain barrier. And so the real question here is how to study what is the environment there and how that environment changes under ocean acidification, and if it changes under acidification or not. And that is when it's like something that is a challenge, is that sampling that fluid is not trivial. And even with fish, many times, if you grab a fish and you take blood samples or a cerebrospinal fluid sample, all the blood chemistry changes due to the stress of the capture, exposure to air, the gills may collapse, anesthesia might affect those parameters as well. So it's not that easy to measure acid-base parameters under physiologically relevant conditions or that reflect the normal biology of the fish. But our colleagues from University of Exeter, Rod Wilson, and students and postdocs, they develop a technique that allows you to perfuse the gills with a benzocaine. It's an anesthetic that doesn't change the blood pH, and then allows you to take blood samples and in some cases also cerebrospinal fluid samples to look at these things. And what we've been doing here is, a lot of this work is by Garfield Kwan, who was a PhD student in my lab. Now he's a postdoc at the University of Exeter. And more recently, well, Trevor as well, but then more recently we started working with Richard Anneman, who's also at ADC NICE, and Ben, who's here in the audience, is also at the ADC NICE and has one of the Kavli Discovery Fellowships that has just been started. And on one hand, what we're trying to see is trying to look at using single nucleus RNA-seq. This is all data that Ben has generated from a variety of species, trying to characterize the cellular genetic or transcriptional profile of many of these cells. Some of the blood-brain barriers might differ or to be different from one species from the next one, and that might hold the key to differential responses. But also, even though I think this is like absolutely very, very exciting, we cannot get away from doing regular physiological measurements and trying to characterize the microenvironments that the neurons might be exposed to. And this is what I'm going to show you next is a great example of the Krog postulate. For any particular problem, there might be an animal better suited for. Because Garfield, he figured out that he could take advantage of lumpfish, which they have a very large skull, and they have a very large fluid, cerebrospinal fluid. And when he looked at the conditions there, just have to rush here a little bit. The story here is that the cerebrospinal conditions in terms of pH, they do not respond in the same way as the blood, and that is, it seems that they be regulating the pH at a different level, probably different mechanisms, and that raises the questions whether different species might be more robust or less at doing this type of regulation. We have similar type of results with otolith function. The take-home message here is that the endolymph, otoliths, they grow larger under ocean acidification, and the key there is that endolymph, the acid-base conditions in that endolymph is different from in the blood in the way that they regulate pH, but by regulating pH at a higher level, they accumulate more bicarbonate and more carbonate, and that changes the biogeochemistry, and that leads to larger otoliths. So whereas under ocean acidification, you have less carbonate in the ocean, in the endolymph, you actually have more. Importance of microenvironments. At the level of the mechanisms, that nothing seems to be happening transcriptionally, so whatever is regulating the pH seems to be more rapid, more dynamic, more post-transcriptionally, which makes it more difficult to study, but also more fascinating. And finally, the questions here are, how are they able, even if you have a larger otolith, is there any room for adaptation of the sensory system to that larger system, to that larger otolith? And also on another scale, whether these biogeochemistry changes in the otolith might affect the rings, which are used by fisheries, and whether that might affect fishery stock assessment and things like that. So this is a problem that starts with neurobiology and physiology and has broader implications. So finally, the take-home messages is that animals live and have evolved in naturally variable environments, and that's something that we need to consider. We are considering already, all of you, so that's great. We have to embrace variability in our results and our experiments, because that is, in large part, what is going to determine adaptation. The idea that many of these animals, the mechanisms are intrinsically different from those from humans, and that requires specific research at all these things. We cannot just base everything on zebrafish or mouse or other things. And that neurons are not directly exposed to the environment, and integrative homeostatic mechanisms are as important as the neurons themselves. I talk about the example of acid-base regulation, but there are ways of delivering more oxygen, or there may be ways of delivering more oxygen, and there are other ways in which the organism might buffer the nervous system from whatever happening in the wild. So how do we study all of this in world animals? That's why we had the Allen Discovery Center for Neurobiology in Changing Environment. Thanks again to the Paul G. Allen Frontiers group. We're trying to do this multidisciplinary approach ranging from genes to the environment. And many of us are here. You heard from Amro and Didi, the two co-leaders. Matt just spoke. Tracy Larson is here as well. Falco Kuster. We have many of the postdocs or posters here, so we're very, very interested in fostering collaborations, trying to help with resources or tools. The only thing we don't have is money, so don't ask us for money. But everything else, we are very happy. We're here until today, tomorrow, and please, talk to us, and we'll be very happy to collaborate. Thank you.
Thank you so much for the talk. And do we have some question for Martin? Yeah, Denise?
Hey, I'm Dennis. I'm a postdoc at OHSU in Portland, Oregon. Great talk. You've shown that the hypercapnic fish are a little scared. Could it be that they just move less? And also, if you give them, let's say, seizure medication, if you boost their GABAergic transmission, can that rescue the phenotype?
Yeah, great question. We look at movement. We don't think that locomotory ability is impaired itself, but some experiments include adding like gabacin or muscimol, and gabacin, it has exactly the effect. It doesn't do anything to the high CO2 fish, but it turns the control fish into anxious, which is what you would actually predict. And then muscimol, which is a pharmacological agent that does the opposite, it reverses the effect. So that there is some mechanistic links with these receptors. Yeah.